Once the sperms are produced in the seminiferous tubule and are released into the lumen, they travel through these duct systems now. Looking at the duct system, first of all, the intratesticular ducts, so still within the testes, the first region they reach are the tubuli, recti. So these are the terminal portion of the seminiferous tubules. They can best be described as series of short, straight tubules which are lined with Sertoli cells only. So in other words, they are losing their spermatogenic cells completely, and now they're becoming lined with Sertoli cells, and then they eventually transitioning to cuboidal epithelium. That is seen in the next stage, referred to as Wheat's testes, and they are the interconnecting channels in the mediastinum, in this region here, circled with the red circle here, and they are lined with the simple cuboidal epithelium. And one of the features that you should note is the presence of now the apical cilium inside here as the sperms are traveling through this region. This is a histological view that you'll be looking at, and you can see in this section there are seminiferous tubules in cross sections. You can see the epithelium with all these different cell types. And then when you come to the end of that, in this case somewhere around here, you can see these rather straight channels, and these are the tubuli recti. So this is where all the cells lining there are the Sertoli cells. As opposed to the Wheat testes, which are now more convoluted, and again in this picture here in the low power, you can still see the seminiferous tubules here, the Wheat testes here, as well as you can start to see part in this high resolution view, some of the tubular recti, which are next to the seminiferous tubule, which will then lead to the Wheat testes. And here in the lumen, you're starting to see some of this material, which is the matured sperms. So having gone through that stage, the next stage is the extratesticular ducts. So one of the regions where this happens is referred to as the efferent ducts, and they are pointed out here in this picture of the testes and the epididymis. So these are the connection between the two, and they are generally between 10 and 20 tubules, which are leading from the Wheat testes to the epididymis. These tubules or ducts are now lined with pseudostratified columnar epithelium with cilia. One thing to note here is that there are clumps of tall and short cells which give rise to a rather wavy appearance in cross-section, and that is highlighted in this picture on the left, where you can see the lumen with sperm inside and this kind of very uneven epithelium, much more easily visible here in even higher magnification. And also you notice the smooth muscle on the outside. So once the sperm has reached that stage, then they end up in the epididymis itself, which is a very highly coiled structure, which can be very lengthy, 4 to 6 meters in length. And once the sperms get to this place, they are pretty much in holding pattern, and that's where the newly produced sperms become mature, and then they acquire their motility. The profile of this tube, as you can see in this high power cross-sectional view, is pseudostratified columnar epithelium with tall cells, which is most of the cells forming here, and some short cells. Short cells are referred to as the basal cells. The tall cells are referred to as the principal cells. The principal cells themselves have stereocilia. The smooth muscle surrounds the duct profile, as you can see in this particular section here. As I said before, the epididymis serves as a holding or a reservoir for the mature sperms. Subsequent to that, the sperms have to go through this another section which is past now the epididymis, and that is referred to as the vas or the ductus deferens. And so this is continuation of the epididymis. And again, this vas deferens is lined with pseudostratified columnar epithelium. And again, it resembles very much like the epididymis itself. However, the lumen of the vas deferens is different to what you see in the epididymis. It is not as smooth as it was in the epididymis, and this irregularity in shape is likely to be due to the fact that vas deferens has very thick muscular coat, and that causes contraction, and then it causes the shape change within the lumen. So near the end of the vas deferens, it enlarges and joins the ducts of the seminal vesicles rather, to form the ejectorate duct, which enters the prostate gland, which we'll see next. So just a overview of this vas deferens. It runs from this region roughly all the way to the prostate, and this is a cross-sectional view. You can see this nice pseudostratified columnar epithelium with the sperms inside, as well as very thick muscular coat. So subsequent to that, the next thing that we need to look at are these accessory sex organs, which is where the sperms are then, as they're traveling through the vas deferens and towards the urethra, these stages are reached. First of them is the seminal vesicles. This is present in a pair. They have a very highly folded structure. The epithelium is again pseudostratified columnar epithelium. One thing to note is that the mucosa here is highly folded, gives the appearance that there are multiple chambers in here. As a matter of fact, there is only one lumen. So it's one tube which is very long, present in a convoluted way. The wall of the seminal vesicle consists of fibroelastic connective tissue and some smooth muscle. The secretory products within here constitute about 70% of the ejaculate from the humans, and it contains fructose. Fructose is the principal metabolic substrate for the sperm, their energy as they're traveling towards the urethra. The smooth muscles here will contract during ejaculation, thus moving this fluid into the ejaculate duct and through the urethra. One thing to note is that in the seminal vesicles, you will not see any sperms. The structure also nearby is the prostate gland. This is about the size of a walnut, has a central lumen, which is the prostatic urethra, which is shown here in this picture, and you will see that in the lab as well. It consists of multiple tubuloalveolar glands and multiple ducts, which open directly into this prostatic urethra. So this is all this being pretty much secretory portion, which then eventually ends up in this prostatic urethra region. The epithelium is typically simple columnar. However, these cells, based on their influence from testosterone, may be cuboidal or squamous or pseudostratified. So there's a variety of morphologies in this area. The other thing to note here is that the alveoli within the prostate contain these what's known as the prostatic concretions, and they would appear as more or less like a concentric circles, and they vary in shape and size, mostly seen in older individuals. These concretions, basically there's no real reason why they're there, but they are sort of like partially calcified, so they actually help you to identify that you are in the prostate gland. Prostate itself does have a dense capsule around it. So the secretions from the prostate glands include the prostatic acid phosphatase, fibrinolysin, citric acid, as well as prostate-specific antigen. And this enzyme is secreted into the prostate gland alveoli, and then it is incorporated into the seminal fluid. Normal individuals have a low level of PSA, but in prostate cancer, the concentration of this could be used as a marker and progression of the disease. So subsequent to this, the next part that we need to look at is the erectile tissue of the penis. The penis itself, it consists of 3 separate cylinders of erectile tissue. In histology lab, we see this erectile tissue mass that contains the penile urethra, known as the corpus spongiosum, and we have the other 2 masses which are located dorsally, and they are called the corposa cavernosa. So each of these cylinders is a collection of irregularly shaped vascular spaces which are lined with vascular epithelium. The erectile tissue around it have tuberculi that contain elastic fibers and some smooth muscles. And the erectile tissue masses are surrounded by a dense fibroelastic layer known as the tunica albuginea, which is highlighted here in this particular section. So in the growth lab, you will study these components a little bit better, but in our sections, we may not be able to see histologically all the components which are shown here in this picture. So this is just a more sort of highlighted view where you can see the corpus spongiosum as well as the penile urethra located within it. So this is what you will be seeing in histology. So in this section here, where you have partial section, you're seeing the corpus spongiosum with the urethra within it. Here, for example, it is lined by pseudostratified columnar epithelium, surrounded by loose connective tissue, as well as you have what we refer to as the glands of Litter, which secrete lubricating stuff into the urethra. This is a section that you're going to be seeing which is less developed because it's an infant penis, but nonetheless you can see the urethra here. You can also see the spongiosum as well as cavernosum here up there, 1 and 2.